Hello students, I Shipra Banerjee from Breno University want to give a lecture on nucleic acid or DNA RNA. You know what is nucleic acid? Sometimes the children having the same faces or same eyes, same hair, same body height is resembling to their parents. You know why? Actually, this is due to the genes or the hereditary concepts. So that means some of the genes or chromosomes that will carry some information from the parent to offspring, and that information is carried through the nucleic acid. That is, in general, you can know as the DNA. So, how the DNA is synthesized, what are the composition, we will be learning through this lecture. So, we can say the nucleic acid is a molecule of heredity. That means that allows organism to transfer genetic information from one generation to the next generation. So this is nucleic acid is composed of generally nucleic acid, some polysaccharide and some protein also. This is the biological polymer of the chromosome or genetic material. So that was discovered first within the nucleus, and that is presence of phosphate group, and that was present in the chloroplast and mitochondria also. But the nucleic acid genetic material is the most important. That was first discovered by F. Peister, one of the scientists. He isolated some DNA from the host shell and he discovered this is the characteristics of some hereditary matter. Now, if we say the DNA, that is a full form of deoxyribonucleic acid, and the RNA, ribonucleic acid. So, from the name, you can understand deoxy. That means removal of the oxygen group from the ribonucleic acid. That means राइबोन्यूक्लिक एसिड जो है उसमें अगर एक ऑक्सीजन हट जाएगा तो उसको हम बोलेंगे डीऑक्सीराइबोन्यूक्लिक एसिड और डीएनए. So this is the monomeric unit of the nucleic acid. So this is composed of nucleoside and nucleotide. So nucleoside is composed of sugar and nitrogenous base, and just 1 second. Generally, this is composed of sugar, then nitrogenous base, which is known as the nucleoside, and one of the phosphate molecule will be coming to tighten this, as well as it will be known as a nucleotide. So these two nitrogenous base is composed of purine and pyrimidine. There are two categories, and from the name you can remember the structure. Purine having the small name, but the structure is small amount of large. It is composed of two fused heterocyclic ring, and pyrimidine, that is name of the large, but the structure is small. So purine having the two types, which is adenine and guanine. They are the two heterocyclic rings are fused. Okay. And pyrimidine, there is one heterocyclic ring is present. So in case of purine, adenine, that is composed of 6 nitrogen compound group, okay, and guanine, and cytosine is composed of 2 nitrogen containing heterocyclic ring, and the position of the oxygen and methyl group and the amino groups are different. So you should remember the structure: cytosine, thymine, and uracil. So this is shortly represented as ATGC, adenosine by adenine, thymine, guanine, and cytosine, and uracil that is present in the RNA that is known as the AUGC. Now, the pentose sugar in RNA and DNA. So this is the ribose sugar present in the RNA. That's why this is structurally present in here. See here the nitrogenous base, adenine or guanine, whatever, or you can say cytosine or uracil. See, this is the structure: pyrimidine, cytosine or thymine or uracil. Anyone from nucleotide will be present, and purine or pyrimidine base. Then the phosphate group is there. This sugar is there. So this sugar moiety gives the rigidity. Okay. A nucleoside, pentose sugar, and nitrogenous base composed of a nucleotide is pentose sugar, nitrogenous base. That's one of the phosphate group has been added here. Okay. So by this way, you have to remember the structure. Next, you can say primary structure of the nucleic acid is like this. Just 1 second. And also. So you can see nucleotides are joined together by phosphodiester linkage between 5' and 3' carbon atom of the pentose sugar to form a typical dinucleotide. So if we see the structure, we can zoom it. So the sugar molecule is composed of 1, 2, 3, 4, 5, and it is started from the 1 carbon to 5 carbon. And 1, 2, 3, this is the third carbon and this is the first carbon. So this compound is composed like this linear way. You can see the structure. So this nitrogenous base is attached to the ribosome or the pentose sugar, whereas this is composed with 1 carbon, and again, and phosphate group is there, then this carbon is attached to the third carbon. Okay. So by going on, the compound will be lengthened. So in case of adenine, cytosine, guanine, and thymine, they are sequentially placed, and the sequence is very beautiful because two purine and pyrimidine. That's why they are linearly placed. And this bond is known as a 3,5-phosphodiester bond because one phosphate group is there, and at the 5 position, one phosphate group is there. Again, in the third position, one phosphate group is there. That's why the chain is started from 5' to 3'. That means one of the 3 carbon will be free at the one end, and one of the 5 carbon will be free at the another end. Okay. This is the free phosphate group, and here you can see the base is attached with the sugar. Okay. Now we can see who are the discoverer of this model. You know, Watson and Crick is the scientist who got the Nobel Prize in 1962 for discovering the structure of the DNA. Okay, so if we can zoom this structure, so DNA is a spiral, what can I say? They have a spiral chain, and they are, whenever this cycle is composed, there are different adenine, guanine, cytosine cells are linked together with the hydrogen bond. Okay, one alternatively, 2 hydrogen bond, then again 3 hydrogen bonds are there. See, hydrogen bond, 3 hydrogen bonds. Adenine, guanine, cytosine. So if you can zoom it, you can see guanine and cytosine. So see, adenine and thymine, 2 hydrogen bond, and cytosine and guanine, there is 3 hydrogen bond. Therewith, alternatively, these two chains are opposite in each direction. So there is no repulsion. As a result, it's having a very stable structure. Adenine and thymine, 2 hydrogen bond, for cytosine and guanine, 3 hydrogen bond is there. So there are 4 types of DNA which can be found. So A DNA, B DNA, C DNA, and this cytogen DNAs are named according to the turn. Okay. Apart from the turning, how they are aligned. Next, I'm going to structure the elaborate. See the structure elaborately. This is the hydrogen bond present in thymine ATC or ATGC. You can see this composition ATGC, GC8. Okay. This phosphate groups are attached. There are 1 millimeter of length in between the two helix or 2 turn. This is a major group, this is a minor. Major group is a expanded and minor group is a small. So from one turn to another turn, 73.4 millimeter length, and in between 2 bases there is 0.34 millimeter length is there. So there are different properties like denaturation, that will be if the hydrogen concentration or the pH or any temperature is increased or decreased, the hydrogen will be broken down the phosphate group. So increase in the absorption of ultraviolet light, that is known as a hypertonic effect, that will destroy the structure. Suppose any cell is kept between UV light, low pH 1 to 3 or high pH above 12, the structure can be found. And the melting temperature of any component can break down the structure. So the spiral helical will be separated out. Then again, if it is cooled, then annealing or denaturation can be done, but that is in a rare case. Now, central dogma. What is central dogma? That is one of the very important term that is proposed by Francis Crick, one of the important scientists. The central dogma for molecular biology is a theory that genetic information flows in single direction, DNA to RNA, DNA to protein. So this is the another name of the process is there. So if from DNA to DNA, that is known as the replication, then DNA to RNA known as the transcription, RNA to protein is known as the translation. So this is known as the central dogma, which is a transfer or flow of genetic material or information from DNA to RNA. Nobel Prize in Psychology and Medicine, 1916. So from India, Hargobind Khorana, one of the scientists, have worked on this. So see the DNA structure. Whenever DNA to DNA will be there, the spiral chain will be linear, and mRNA will be there. There are different mRNA, ATCG. So this mRNA forms different codon, and from this codon, different hormone can be there, different can be there, or amino acid can be there, like methionine, isoleucine, serine. These are the important names of different acids. Sure. Now coming to the function of DNA, you know very well. DNA has the roles, three important functions. Storing information. Genes are segments of DNA that carry messages to make protein, like the good hereditary, bacterial hereditary, everything. Copying information so that when cells divide, all cells get a complete copy of genetic material. That means whatever the property is there, that will be equally divided into cells. And that's why it is said that every person having an identical person in all over the world, maybe it's known to her or him, or maybe it's unknown to her or him. That's why it has been many times found that in cinema we can see the twins are found. Okay. Same type of thing. Because this copy of this another genetic material is transferred to from parent to parent, parent to parent. Transmitting information. DNA is passed from parent to parent. As a result, information is passed. Now there are different type of RNA also there. That is one mRNA, tRNA, rRNA. mRNA means messenger RNA that carries the message from DNA into the cytoplasm of the cell. Transfer RNA that carries amino acid to the site of synthesis. rRNA that is the facilitating binding of mRNA to the ribosome. So next, this is the again structure of RNA has been found. This is not the spiral chain and not the two pair of strand is there. That's why one pair of adenine cytosine is there and one of the phosphate is there in the ribosomes or ribosome. This is a single stranded. RNA is single stranded. Now again it is detailedly known as what is RNA. RNA is a nucleic acid called ribonucleic acid. Then the function of RNA like the RNA transfer genetic information from the nucleus to cells and make up ribosome and help assembly. They are the property of the RNA. Okay. So this without RNA protein synthesis cannot be there. So this is a very brief presentation of DNA and RNA and the nucleic acid. Hope you have understood.